A little bit similar to what David showed you, I'll be going through this example using simulated data. I'm going to do, the Spec2 pipeline, I'll do it in two passes. One, just running the pipeline in full by itself so that you can see what typical data products look like for that. And then I'll go back and do a second pass running some of the individual steps. That way you can get a better feel for what they're actually doing to the data. And then I'll end with a pass of the full Spec3 pipeline and the products that produces. So for this, really, it's intended to show you more from the data point of view, what's going on as you process data through the various stages. As David noted, again, for this notebook, using the pipeline version 1.1.0, and these runs here will be using all the default reference files that are used for that build. So just to keep in mind that the pipeline is in continuous development. So there are new changes being made all the time. So just note that what you'll be seeing here isn't necessarily the most in-date version of what you might actually see, and that there are some caveats to be aware of. In particular, because we're using a lot of reference files for the various steps that are generated either from limited ground-based data or simply are placeholders because we can't actually measure them until we get on orbit. That affects sort of the pixel values that you see after processing. So in particular, you have to take, particularly the flux information per pixel with a grain of salt. So for the simulated data that I'll be showing, these were generated by ESA's Instrument Performance Simulator using an input scene that was a point source using a model PSF and then a stellar spectrum taken from a model of an A star. The observation consists of a total of 9 exposures taken through the S200A1 slit, one of the 5 fixed slits. Using a dither pattern, including the 3-point primary dither, where the source is moved down the length of the slit to enable, the main point for that is to enable subtraction of nodes for background subtraction. And then also an additional spectral sub-dither, where at each of those primary positions, the source is moved slightly to either side of the slit, along the slit width in the dispersion direction, with the intent to help improve the sampling, the spectral sampling. The instrument for this case was configured with the prism disperser and a detector subarray that's the standard subarray for this particular slit, the sub S200A1, which is a 2048 by 64 pixels on each detector. In this case, because we're using the prism, that's fully imaged onto only one of the detectors, so we'll only be including one file per exposure for that particular detector. If you were using other dispersers, you in general would see 2 files corresponding to the 2 detectors for each exposure. I've named the files here for convenience to have a short name. They identified according to their position in the dither pattern. It's not what you would see in the archive. There's a standardized naming scheme for that, but I figured that wasn't as helpful for this, so we've renamed the files this way. Here is the dither scheme, which basically shows you a schematic of where the positions are on the slit. So in addition to the caveats with the pipeline, there are also some caveats to be aware of for the simulations. I don't have time to go into this in detail, but the main point here is that the simulator doesn't include all of the effects that are treated by the pipeline, in particular some of the throughput effects that are treated in the flat field step. Just to note that, again, take the flux information, don't take it literally. This is just an example just to sort of demonstrate the kinds of products that you would see. So I'll start off by importing some packages that you need. So I'm just going to shift enter through these cells. some packages for data visualization and plotting using matplotlib. And then, as David showed, loading all of the modules needed for the pipelines, as well as some of the individual steps that we'll be going through. Then I have specified an output directory to sort of organize the data. This is will be used when we run the pipeline and individual steps. The output products that are generated will be placed into that particular output directory. Then we have a plotting routine there. And now we'll start by downloading the data. So I've put all of the 9 exposures, 9 FITS files corresponding to the exposures on box in a zip file. These cells will download that zip file and unload all of the files into the local directory. And so we'll start off just by showing you what this input data looks like. Now, one thing to note, the simulator products are generally electron rate maps. It's usually not able to generate raw up the ramp data, so we won't be able to run the stage 1 pipeline with these simulated data products. We'll be starting with the stage 2 processing. And so the inputs here are basically equivalent to the level 2A count rate data products, where we've basically packaged and reorganized the information to be in a format that's compatible with the pipeline with all of the proper header keywords. So let's look at a few examples just to show you what these look like. This is for the first dither position count rate image. Here you can see the subarray, 2048 by 64 pixels. And you can see here towards the center, sort of in white, that's the spectral trace of this point source. This is for the prism disperser, which is the lowest resolution disperser available for NIRSpec. So it produces a relatively short trace fully contained within the subarray on a single detector. And if we look at one of the other dither positions, dither position 4, you'll see something similar, but you'll see the source traces moved down in the subarray as a result of that dither position being lower, further down on the slit. So let's move on to processing these data through the Spec2 pipeline. I'm going to do this using association files. You've already seen some explanations about what those are and what they look like. Here I'm using some examples of what you would actually see from the archive from the automated pipeline processing of the data. For Spec2, there's basically one association file per exposure that lists at minimum that particular science exposure. In general, there's a few use cases for associations at level 2. Primarily, that's to enable background subtraction. For example, in this particular example where we have 9 dithered exposures, where some of the dithers are used specifically for background subtraction, those elements are actually specified in the association files. And when the pipeline gets to the background subtraction step, it will read the contents of the association file and determine which are to be used for background and which should get subtracted from which of the science exposures. So here's an example of the contents of one of these association files, where you can see here listed, this is for the science exposure that's dither position 1, and then here is the full set of exposures to be used as background. Basically, all of the exposures at the second and third primary dither positions. So now we'll look at the run of the Spec2 pipeline. So here I've commented this out because it takes a little bit too long to run this in real time, but I've pre-computed the products here, but this is just to show you how to run it. If you want to run this yourself offline, you can do that. Simply uncomment all the lines in this cell. But this is basically shows you how to invoke an instance of the full Spec2 pipeline, how to save the results. So I'm skipping the flat field step here because of the limitation of the simulations not including all of the components that are treated by the flat field. Later on, when we go through some of the step-by-step processing, I'll show you an example of what that looks like. So we're not going to run that in real time. Here is the full set of products that you get from running the full Spec2 pipeline. This is basically looping through all of the exposures and displaying the contents of the, basically the contents from the SCI extension of the FITS files for each of the products. So, for example, here starting with the dither1 position, this first plot shows you the calibrated 2D unrectified spectrum. This is the primary product generated by the Spec2 pipeline. Here, since we've run background subtraction, including all of the appropriate background elements, you can see the negative traces that are imprinted on the data after that subtraction, and then the positive trace. That's the source for this particular science exposure. And then for the quick look products, for this exposure level data, we have the 2D rectified spectrum, where this has been resampled onto a rectified regular grid in spatial and wavelength coordinates. Here, because we're looking at the prism disperser, where there's very little curvature of the data, you don't see much of a difference in these, but there is actual rectification going on. And then finally, a quick look 1D spectrum that's extracted from this rectified 2D spectrum here using extraction aperture centered at the position of the source here. And then you can browse at your leisure the other exposures in this dither set. Well, you'll see once you move to different node positions, you'll see the position of the positive source trace change. So now let's go back and run through again step by step, just to show you what's happening in these particular steps. So here I'm just going to, for speed's sake, work with a single exposure, which is the dither position where the source is in the middle, at the very center of the slit. So first we'll run the assign WCS step. As has already been described, this essentially takes, for NIRSpec, we actually use a parametric optical model of the instrument where we calculate transforms between various optical planes interior to the instrument. And the result of all those transforms is calibrated and encoded by this step into the ASDIF extension of the output FITS file. You can access all of these transforms by means of the WCS object that's available there. So in this cell here, I'm going to load the data model for the output of this step, and then access the WCS object like this. And so here, for example, I list, similar to what David showed for MIRI MRS, you can see all of the various planes that are available for computing transforms between any two of these. For example, the detector frame, various frames within the instrument, different optical planes in the instruments, such as the grating wheel, the Slit and MSA frames, and then also various frames on the sky, going all the way out to world coordinates, essentially RA and DEC on the sky. And this here shows an example of the units for some of these frames. For example, the detector is in pixels. You can transform from pixel frame in the detector, for example, all the way out to sky. In a few steps, I'll show you an example of how you can use these transforms to get some specific information of the WCS coordinates for each pixel in the 2D spectrum. The next step is, this is the background subtraction. So this, if there are any background elements in an exposure set identified as backgrounds associated with the science exposure, they will get included here. If there are multiple backgrounds, they'll be averaged together and then subtracted from the science exposure. Typically, again, as I showed you with the full Spec2 run, you would specify these in an association file. Here, since we're running an individual step on a single science exposure, I'm inputting these using a list, including just two of the other exposures in this set. And so basically those two exposures get averaged together and then subtracted from the science exposure. And here's what the result looks like. So you can see here the dither position that I used where the source is centered in the slit, you can see here as the positive trace, and then the negative traces because of the background subtraction are apparent here. The next step is extract 2D. So this will use the WCS information determined in the assign WCS step, determine the bounding box within which contains all of the usable data that's appropriate for this given aperture. So in this example here, you can see the prism only disperses into a relatively small range of all the pixels available in the subarray. Extract 2D will basically zoom in on this usable range. It doesn't really do a whole lot other than that for single object modes. This is most important for the MOS mode, where we need to extract all of the multiple sources that are present within a single exposure and allow us to calibrate those individually. But here I'll just run this really quick. Here you can see in the verbose output will actually tell you what the bounding box limits are in X and Y. And then here's what the output is. You can see, again, similar to above, but now we've zoomed into to just essentially the usable region. Okay, now using this output, here is where I'll show you an example of how to use the WCS information. Here we're going to try to plot where the trace of the source is in the 2D spectrum using the known source sky coordinates, the coordinates of which are actually present in the header of the data. So this code basically will be starting from pixel space in the detector frame, so XY indices of the pixels in the 2D spectrum, then get the transform that allows you to convert pixels in the detector frame to values in the world coordinate frame, or array-decant wavelength, basically, is the output in this case. And so here is what actually affects that transform and gives you those values for each of the detector pixels. And then we take the source coordinates from the header keywords here, and then determine the trace of that on the spectrum, and that's shown here. You can see the red and purple lines are both exactly on top of each other, so you're only seeing the red here, showing you where the array and deck position of the source is located. It nicely overlaps with the actual source trace here. You can also use this in the wavelength direction. It's probably where it would be most useful, particularly if you had an extended source and wanted to see where, for example, an emission line of interest might be located in 2D space. You can use the transforms in a similar way to find that out and display that. The next step is the source type. In this case, most of the steps that follow this depend both in an algorithmic sense and also in terms of what the wavelength per pixel is. It depends on whether the source is a point source or whether it's an extended source. And so this step makes that determination. Right now, it's only for the fixed slit mode, it's only determining that based on a header keyword that is taken from information from the PPS database. When you design an observation with fixed slit mode in APT, you have the option of specifying whether the source is point or extended. If you make that determination there, that information gets passed down through the data management system and gets incorporated into the data. This step of the pipeline will look for that header keyword and make a determination based on that. In this case, because we're using simulated data where we haven't gone through that full end-to-end process, the keyword is not populated, it's set to unknown. And in that case, the pipeline for fixed slit mode will default to point source. And so source type here should be set to point in this case. And then in this cell, we'll actually print out the keyword that's populated by this step, and we see indeed that's set to point. The next step is wavecore. So for point sources, when a source is not positioned exactly at the center of the slit in the dispersion direction, the wavelength scale needs to be adjusted to account for the shifts in that. It's a wavelength dependent correction. it's not just a simple scalar shift. And so this step will take that into account, knowing where the point source is located in a relative sense within the slit. That correction then gets applied. In this case, since I'm using a dither position where the source is in the center of the slit, that correction is essentially 1 here, so there's nothing terribly interesting to see. The next step is flat field. So now I'm actually going to run this, even though there's not a full treatment of it in the simulated data, but just to show you what this actually looks like. For NIRSpec, this is a complicated step. We have there's 3 components that are combined through this, treating different areas of sort of throughput-related information. So one is the D flat, which covers the pixel-to-pixel response variations at the detector level. Then there is the S flat, which includes all of the throughput losses that are incurred in the spectrographic stage of the instrument. So that includes the grading wheel and optics that image onto the detector. And then finally, there's the F flat, which folds together throughput of the filter, 4 optics, and the OTE. So each of these components we have to calculate on the fly because they are wavelength dependent, and so we need to actually have what the wavelength values for each pixel in the 2D spectrum, which is determined by the assigned WCS step. Take that information, run through the reference files for each of the components, and do interpolation with wavelength and compute these values on the fly. And then the values for each of the 3 components are then multiplied together to give the final set of correction values. When you run this step, you have the option of outputting a 2D array of these correction values, the combined correction values. And that's what I'm going to do here in this example, running this step. And then here is what that looks like. This takes a little bit of time to compute the corrections. And then here you can see, here's the 2D array of correction values. These are all the pixels that receive light from this particular slit. And all the correction values have been combined together here. And then this basically gets divided into the science data to then generate the final flat corrected 2D spectrum. The next step is path loss. So what this does is account for cases where when there's a point source, if that source is not located exactly in the center of an aperture, a fixed slit or an MSA shutter, a correction needs to be applied to account for the difference in the slit loss between an off-centered source and the centered source case, and that's what's done here. This is also a wavelength-dependent correction, so it requires, again, the wavelength values per pixel. And so I'll run through here. Again, because I'm using a source that's centered in the slit, the correction in this case is essentially 1, so nothing terribly interesting to see. Then we have the photom step. So in here, since we've already corrected for most of the throughput terms in the flat field step, all that this step is really doing here is then applying a final conversion factor that converts the data units per pixel from corrected data numbers per second to actual physical units. In the case of point sources, that's megajanskis. If it's an extended source, the units will be megajanskis per steradian. In this case, since it's a point source, we should see the photometry keywords. And here are the keyword values that you would get from this, here showing you what those conversions are. This is another case where currently we have only placeholder reference files where the conversions are just 1. So the units you'll see will look kind of funny with final flux units because of that fact. It'll look kind of weird. Once we get on orbit and are actually able to observe calibration star standards, we'll actually determine real calibration factors as part of this step. And also, there's an option here for this to have a wavelength-dependent vector in case any of the previous throughput corrections aren't able to fully correct for all of the throughput terms. That sort of residual correction can be folded into this step as well. Then we have resample spec, which takes the calibrated 2D spectrum and then resamples it onto a regularized grid in spatial and wavelength coordinates. And then that just shows you that example there, rectified. And then finally, extract 1D will apply an extraction aperture to this image and sum up as a function of wavelength and produce a 1D spectrum from that. And here is what that looks like for this particular exposure. Now, I just have a few more minutes, so I'll try to run through this really quickly for the Spec 3 pipeline now. So this is intended to gather all of the exposures obtained in this dither set and then combine them together, shifting all of the different dither positions so that they all overlap and provide you with a combined spectrum in the end. Here we use an association valve, as David already showed you what that looks like. For the MIRI MRS, a similar thing here. Association file just lists all of the cal.fits files generated from the Spec 2 pipeline run and then feeds that into the Spec 3 pipeline. Here I'm skipping the outlier detection step because the simulations in this particular case didn't include a full treatment of the noise, and that doesn't work well with how that step runs. So we need to skip that step. Otherwise, you'll see a lot of incorrectly flagged pixels. And then I'll scroll down here, and here is what the final products look like. Here's the combined 2D rectified data where you can see negative traces from the background subtraction, and then the final overlapping trace of the source itself seen there. There's an extension part of that product that actually shows you enables you to look graphically at what the overlap between all of the input exposures actually looks like, shown here. And then finally, the combined extracted spectrum from that I plot here. In the interest of time, I'll end here. The remaining cells of a notebook actually shows you an example of how you would rerun the extract 1D using a different extraction aperture, just to give you a demonstration of what that would look like, how you would actually go modify a reference file for a particular step and then rerun to get a different